The Flow of Genetics information kit is a kit that's meant to help students understand the steps of the Central Dogma. The Central Dogma starts off with DNA replication. After the DNA is replicated, it moves on to transcription, the process of turning DNA into RNA. And finally, translation, the process of RNA becoming a protein. No, DNA doesn't literally turn into RNA, and RNA doesn't literally become a protein, but it's just through these processes where RNA and protein are made.
The Central Dogma begins with the process of DNA replication. DNA replication is the process of using a pre-existing strand of DNA to make two brand new strands of DNA. In the beginning of the process, DNA is unwound by an enzyme called helicase. The helicase then unzips the DNA to create two separate strands of DNA. The two strands of DNA are now called the leading strand and the lagging strand. The leading strand is in the direction 3 to 5 prime, and the lagging strand is in the direction 5 to 3 prime. The process that these two strands go through is different because the strands are anti-parallel to one another, so they will be replicated in different directions. We will begin with the leading strand. An enzyme called primase binds to the leading strand and adds an RNA molecule called a primer, which marks the spot of the DNA where DNA polymerase is supposed to bind to this DNA strand. After the RNA polymerase binds to the leading strand, it reads the leading strand one base at a time by walking along the leading strand and binding each base with a complementary base pair. The leading strand is being replicated continuously in the 5 to 3 prime direction. The process is different for the lagging strand, as multiple primers need to bind to the strand, and because of the DNA polymerase is constantly reading small portions of the strand and then detaching. The lagging strand is being replicated discontinuously in small fragments, which is called Okazaki fragments, in the 3 to 5 prime direction. This is why the DNA replicates semi-discontinuous. After most bases have been paired with one another, another enzyme called exonuclease removes the primer after the lagging strand and replaces it with the matching base for that section of the DNA. After the DNA ligase seals up the two strands of DNA, this process is called semi-conservative because the DNA is made from one strand of the original DNA molecule and one new strand. After replication, it automatically winds up into a double helix. The result is that you end up with two new strands of DNA. If there is a change in the DNA code, then it can be corrected, but if not, then it can cause mutations which can affect the way a protein will be made.
Transcription is the second stage of the Central Dogma, where DNA is transcribed into mRNA. It occurs in the nucleus of a cell, and there are 3 stages of transcription: initiation, elongation, and termination. Initiation is the first stage where the transcription process is initiated. To initiate transcription, transcription factors in the RNA polymerase bind to the promoter, also known as a TATA box. Transcription factors are protein enzymes that initiate and regulate the rate of transcription of genetic information from DNA to messenger RNA. These factors have high affinity to the TATA box. So they come, and they go, and sit on the TATA box. And then, once all the transcription factors are bound to the promoter, which is the TATA box, the RNA polymerase arrives. RNA polymerase is an enzyme that copies the DNA into messenger RNA. Once it binds to the promoter region, the transcription factors will leave. And transcription is initiated. And the second stage of transcription, elongation, can begin. During elongation, RNA polymerase unzips a section of DNA and moves along the non-coding or template strand of DNA. As it moves along the strand, it reads each individual nucleotide base and then adds the complementary base pair to a single strand of messenger RNA that is being created. As you can see, the strand was read and transcribed in a direction of 3' to 5', so that once it's finished, this mRNA strand now looks almost identical to the coding strand of DNA. The only difference is that on the mRNA strand, instead of thymine, there will be uracil. The last stage of transcription is termination. Once RNA polymerase transcribes a sequence of DNA known as the terminator, it knows to stop transcribing. A protein that helps in this process is a transcription repressor. This will be a repressor. This protein binds to enhancers to shut down transcription. Once transcription is shut down, the messenger RNA leaves the nucleus to go find a ribosome to be translated, and it leaves.
Translation is the process of turning the RNA into linked amino acids which become proteins. This is the last step of the central dogma and occurs in the cytoplasm of a cell. There are 3 stages to translation: initiation, elongation, and termination. In initiation, the ribosome is brought together with the messenger DNA and transfer RNA begins translation. This starts when the transfer RNA detects the start codon, which is AUG. The start codon is bound to the P site of the small ribosomal unit, and the initiation transfer RNA base pairs with the AUG codon. A codon is a group of 3 amino nitrogenous acids found in the messenger RNA. Before the start codon, there are a few extra nucleotides on the mRNA sequence. These form a cap that protects the start codon. The small ribosomal unit and the large ribosomal unit will join together to form a functioning ribosome. The ribosomes have 3 sites that transfer RNA binds to. The P site holds the transfer RNA that carries the growing polypeptide chain. The A site holds transfer RNA that carries the next amino acid that has to be added to the polypeptide chain, and the E site is where the discharged transferred RNA leaves the ribosome. The messenger RNA carries the genetic information for the ribosomes, which is made by ribosomal RNA, and the transfer RNA transfers the amino acids to the ribosome where proteins are synthesized. In elongation, the anticodon of another transfer RNA base pairs with the messenger RNA. The ribosomal RNA that is found in the large ribosomal unit catalyzes the formation of a peptide bond, which is a chemical bond that links amino acids, that is between the amino groups of amino acids in the A site. They also catalyze the carboxyl end of the amino acid in the P site. The messenger RNA is translated in one direction, 5' to 3'. Now proteins are made in the N-terminal and the C-terminal direction. Lastly, there is termination. In the last step, the developing polypeptide will leave the ribosome through the large ribosomal unit that has an opening. Before the last codon is reached, the cytosine tRNA is reused for the last amino acid. Finally, the stop codon is reached, and the finished polypeptide is then released into the body and after it is edited, so it can do its job. If the wrong amino acid is attached or proteins are missing, it will cause mutation. One thing that a lack of proteins can cause is lactose intolerance. This occurs when the body doesn't make enough protein.